Hi everyone, myself Swati Priya AK, tutor, Department of Physiology, Sree Balaji Medical College and Hospital, Chennai. Today I am here to give a lecture on passive transport. So at the end of this lecture, you will learn about the transport across cell membrane, how transport across cell membrane has been classified, what is passive transport, how different types of passive transport mechanism is happening like simple diffusion, facilitated diffusion and osmosis with examples. First, we'll see why transport across cell membrane is important. Nutrient uptake. Cell needs essential molecules like glucose and amino acids for metabolism and energy production. Next is the waste removal. As a result of cell metabolism, waste products need to be eliminated to prevent toxicity. Next is maintaining homeostasis. The cell membrane helps regulate the internal environment of the cell by controlling the movement of substances in and out of the cell. This maintains a balance of ions which is essential for the nerve and muscle conduction. Next is cell communication. So cell signaling, in cell signaling, membrane receptors play a vital role which allows them to communicate with each other and coordinate activities. Finally, the cellular processes like cellular respiration also relies on the transport of molecules across the cell membrane. For example, electron transport chain of mitochondria relies on membrane transport for energy production. Now, we'll see how cell transport has been classified. It has been broadly classified into passive transport and active transport. Under passive transport, it is subdivided into simple diffusion, osmosis, and facilitated diffusion. Coming on to the active transport is further divided into transport through membrane pumps and transport through vesicles. Membrane pumps are further divided into primary active transport and secondary active transport. Secondary active transport is further divided into co-transport and counter-transport. Under vesicular transport, it is divided into exocytosis and endocytosis. Endocytosis is of three types: pinocytosis, phagocytosis, and receptor-mediated endocytosis. So this picture is an example of active transport. This shows sodium-potassium pump, which plays a very important role in the primary active transport. In primary active transport, molecules or ions move against the concentration gradient. Thus, it requires energy for the movement of molecules in and out of the cell, which means the molecules will be moved from an area of lower concentration to the area of higher concentration. In primary active transport, the sodium-potassium pump allows sodium ions using the energy of ATP hydrolysis to establish a concentration gradient of the sodium. Then in the secondary active transport, sodium moving along the concentration gradient established by the sodium-potassium pump will drive the force required for the transport of glucose against its concentration gradient. So examples of primary active transport are sodium-potassium pump, or otherwise known as sodium-potassium ATPase pump. Secondary active transport is further divided into co-transport and counter-transport. Under co-transport, examples of sodium and glucose transporter. Under counter-transport, examples are sodium and hydrogen transporter. Next, we'll see about exocytosis and endocytosis, vesicular transports. Exocytosis is nothing but movement of substances from internal of the cell to the outside. Here, vesicles helps in the transport of molecules or substances from internal of the cell. It moves and it attaches to the membrane. Then by exocytosis, exo means outside, cytosis is outside of the cell. The substances are excreted out of the cell, or it is released or secreted out of the cell membrane. Moving on to the endocytosis. Endocytosis is nothing but endo is internalizing. So substances from outside, that is from the extracellular space, are internalized by formation of invagination in the cell membrane. Finally, the invaginated membrane pinches off like a vesicle and it carries the substance which has to be transported to the cell. Endocytosis is of three types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So this is a small introduction about active transport. Now we will see in detail about the passive transport across cell membrane. It is the movement of molecules through the cell membrane along the concentration gradient without any energy expenditure. As the molecules are going to move from an area of higher concentration to the area of lower concentration, this mechanism, this passive transport mechanism, does not require any energy for the transport. It is of three types. First one is simple diffusion, facilitated diffusion, and osmosis. When you look at this picture, you're able to see lipid bilayer. Everybody knows that cell membrane is made up of lipid bilayer. So diffusion is going to happen through the lipid bilayer. Molecules which are easily soluble through the lipid bilayer will easily enter into the cell or out of the cell by dissolving in the lipid molecules or small spaces present between the lipid bilayer. But molecules which are not able to dissolve, that is lipid insoluble or water insoluble substances, are transported through special transport which are called carrier protein or channel proteins. First, we will see about simple diffusion. Simple diffusion, the movement of molecules or ions occurs through a membrane opening or through intermolecular spaces without interaction with the carrier protein. Everybody knows what diffusion is. Diffusion is nothing but movement of molecules from an area of higher concentration to the area of lower concentration through a semi-permeable membrane. The movement of molecules occurs according to the Fick's law of diffusion, which means rate of diffusion is directly proportional to the particle's concentration gradient. J is equal to minus D into A C1 minus C2 by T. J is the rate of diffusion, D is the diffusion coefficient, A is the cross-sectional area, C1 minus C2 is the concentration gradient, T is the thickness of the membrane. So simple diffusion of lipid-soluble molecules, for example, oxygen, nitrogen, carbon dioxide, alcohol, steroid hormones, these are lipid-soluble substances. So they can easily be dissolved in the lipid bilayer and can be transported in and out of the cell. So the rate of diffusion is directly proportional to the solubility of the molecules in the lipid. Moving on to the next one, simple diffusion of lipid and water insoluble molecules. So when they are not soluble in the lipid bilayer, they use a special transport which is called protein channels. Protein channels are selectively permeable, or they are provided with the gating mechanism. Protein channels are selectively permeable, which means it allows only specific ions to pass through them. Gating of protein channels are divided into three types: voltage gated, chemical gated channels, mechanical gated channels. Before moving on to the voltage gated channels, we will see a small introduction about how voltage gated channels are developed. Patch clamp technique. It is a lab technique in electrophysiology that allows the study of single or multiple ion channels in the cell. It was developed by Irwin Neher and Bert Sackman in 1976 and in early 1980s. It is a refinement of voltage clamp. This discovery has made it possible to record the current of single ion channels for the first time and their fundamental involvement in generation of action potential. So it is a useful tool in this. A glass micropipette is inserted into the cell membrane and it is made as a patch. A tight seal is made in which the patch of the cell membrane will be controlled and the movement of voltage across the membrane will be controlled. So we'll be calling it as voltage clamp. So with this tight seal of the membrane, we'll be able to control the current passing through the channels. It is used to study how ion channels open and close and respond to different stimuli like change in the voltage across the membrane. Moving on to the simple diffusion back, the left side picture depicts the channel protein. This channel protein will allow only specific ions, that is selectively permeable, which means sodium channels will allow only sodium ions to pass through, potassium channels will allow only potassium channels to pass through. It depends upon the size and pore size of the molecule in the channel. Next picture shows the gating, voltage gating of the channels. First we'll see the sodium gated channels and then we'll see about the potassium gated channels. The voltage gated channels respond to the electrical potential across the cell membrane. First is sodium gated channels. They are located at the outside end of the channels. Gates are nothing but small extensions, gate-like extensions of the transport protein molecule which can close the opening of the channel or can be lifted away from the opening by a conformational change in the shape of the protein molecule. This opening and closing of the gates are controlled by three types which we have already seen: voltage gated, ligand gated and mechanical gated. Here first we are seeing the voltage gated. In this the sodium gates have sodium channels have gates which are present in the outer end of the channels. So these gates are kept tightly closed when there is a strong negative charge on the inside of the membrane. When the inside of the membrane loses the negative charge, the gate opens and it allows the sodium ions to enter into it. So this is responsible for the basis of the occurrence of action potential for nerve signals. Next is potassium gated channels. Potassium gated channels, in this the gates are present on the inner side of the channel and they also open when the internal membrane loses its negative charge. But Potassium channels will act much slower when compared with the sodium channels. That is why it is partly responsible for the termination of action potential. Ligand-gated channels are nothing but gates of the channel will open when a ligand or a chemical molecule attaches to it. Examples are acetylcholine channels, which play an important role in the transmission of signals from the nerve to the muscle in neuromuscular junction. Mechanical gated channels will open due to the mechanical stretch. These mechanosensitive channels play an important role in the cell movements. Next, we'll see about the facilitated diffusion. Facilitated diffusion, also known as carrier-mediated diffusion. Substances are transported through a carrier protein in this mechanism. Glucose molecules and most of the amino acids are most commonly transported through this diffusion. In this transport, the molecules come and bind to the binding point present in the carrier protein, and then after the binding, it causes conformational change in the structure of the carrier protein, which allows the diffusion of molecule in and out of through the cell membrane. Here, the rate of diffusion will increase according, or it is directly proportional to the movement. Rate of diffusion is directly proportional to the concentration of diffusing substances, but it reaches to a saturation point which is called Vmax, above which the rate of diffusion stops or no diffusion occurs above that. So in this graph, we will be able to see the rate of diffusion and concentration of substances, how they are linked in simple diffusion and facilitated diffusion. So if you see the blue line in the graph, it shows sharp increase, which means rate of diffusion is directly proportional to the concentration of substances in simple diffusion. But when moving on to the facilitated diffusion, the rate of diffusion initially increases, but it reaches a plateau, which means it reaches a saturation point, which is Vmax, above which the rate of diffusion does not occur or diffusion completely stops. Next, we'll see about the factors affecting the net rate of diffusion. First is the cell membrane permeability. Under this, thickness of the membrane, lipid solubility, distribution of protein channels, temperature, size of the molecules, area of the membrane, all these are responsible for the cell membrane permeability. Next is the concentration gradient. So according to the concentration gradient, the diffusion of the molecules will be affected. Next, electrical potential gradient. Membrane potential plays a very important role in electrical potential gradient. Pressure gradient. When the pressure is increased, more number of molecules will be striking the membrane. Hence, there will be increase in the net rate of diffusion. Now, moving on to the third one, which is under passive transport, is osmosis. Everybody knows what osmosis is. Osmosis is nothing but movement of solvent molecules from the area of lower solute concentration to the area of higher solute concentration through a semi-permeable membrane. This is a classic demonstration of osmosis, in which when you see the second picture, the solvent molecule, that is water, has transported from one limb to the other because the solute concentration is higher in the other limb. Now we'll see what is osmotic pressure. Osmotic pressure in the body fluids is defined as the pressure exerted by solutes dissolved in the water or any other solvent. Osmotic pressure exerted by the colloidal substances in the body is called as colloidal osmotic pressure. Oncotic pressure is the pressure exerted by the plasma proteins. The minimum pressure when applied on the side of the higher solute concentration will be preventing osmosis. This is called osmotic pressure. Oncotic pressure, it prevents the accumulation of water in the interstitial fluid. This pressure is responsible for the reabsorption of fluid back to the venous end of the capillaries during the capillary exchange. But when the oncotic pressure decreases, it causes edema. Next, we'll see some important terms like osmolality and osmolarity. The osmotic pressure depends on the number of molecules or ions dissolved in the solvent. The concentration of osmotically active particles is expressed in osmoles or milliosmoles, which is 1/1000th of 1 osmole. Osmolality is defined as the number of osmoles or osmotically active particles per kg of the solution. It is expressed as osmole per kg. Osmolarity is number of osmoles or osmotically active particles dissolved in per liter of the solution. It is expressed as osmole per liter. Next, we will see what is tonicity. Osmolality of a solution relative to plasma will be called as tonicity. Solution with the same osmolality of that of the plasma is called isotonic solution. Solution with greater osmolality is called hypertonic, and the solution with lesser osmolality is called hypotonic. The normal plasma osmolality is 290 milliosmol per kg, out of which sodium, chloride and bicarbonate will be contributing to about 270 milliosmol per kg. Glucose and urea will contribute to about 20 milliosmol, and then proteins will be contributing to 2 milliosmoles. This picture shows when the interstitial fluid is isotonic, what happens when the interstitial fluid becomes hypotonic, and what happens when it becomes hypertonic. So the red cell membrane, the erythrocyte, when it is surrounded by the interstitial fluid, which has the same concentration as that of the cytosol, there won't be any net movement of molecules. When the red blood cells are present in the interstitial fluid, which has less concentration than that of the cytosol, water molecules move from the outer from the interstitial fluid into the erythrocyte, and it swells, and finally hemolysis occurs. In hypertonic solution, that is when the interstitial fluid is more concentrated than that of the red blood cell cytosol, what happens here is the red cell molecules, water molecules from the red cells will diffuse out of it, and the crenation, that is shrinkage of the red cell membrane, occurs. Tonicity is crucial for maintaining the proper shape and function of the red cell membrane. The flow of water in and out of the capillaries depends on whether the colloidal osmotic pressure or oncotic pressure is greater than or less than the hydrostatic pressure of the blood. So the clinical significance are the total plasma osmolality is important in assessing dehydration, overhydration, and fluid electrolyte abnormality. Now, finally, we are coming to the summary of the session. So, we have seen about the transport across cell membrane, different types of classification of transport across cell membrane like active transport with few examples, and we have seen in detail about the passive transport, that is simple diffusion, facilitated diffusion, and osmosis with respective examples, and what is the importance of transport across cell membrane and why it is very essential for our body to maintain proper homeostasis. Thank you.